Good morning, everyone. Thank you for making it through this weather. Morning, Anna. Hello, everyone in 10 Evans. Since I am going to give everyone another minute to get here, so I wanted to share a bit of amazing trivia I learned last night. Don't ask me why. This way of depicting data, 90% of you wish you were still asleep, 2% wants to be here, and 8% wants to be awake, but somewhere else other than here. Who invented the pie chart? Anybody know? Bravo. Florence Nightingale. Do you want to say briefly who she is? Okay. Florence Nightingale is an English woman who is the founder of nursing. There wasn't an institute of nurses, so soldiers would just die in the battlefield. And Florence Nightingale, this remarkable woman, this was during, I think, the 1860s or 70s, she decided that she needs to communicate to the members of parliament in a simple and accessible way that there's a problem. And so she invented the pie chart because it was simple to understand. Why am I telling you this? For no particular reason. Hello. Today you shall get, as promised, mitosis, or a tale with threads from a Berkeley salamander, specifically not the entertainingly named Batrachoceps diabolicus, but the Batrachoceps attenuatus. What exactly does a salamander have to do with mitosis? Everything, as you shall see in a second. Before we dive into mitotic mechanics, I feel obliged, of course, to connect us very briefly to what we spent a couple of lectures discussing, the replication of DNA. I want to emphasize really, really strongly that the goal of DNA replication is not DNA replication. It is the replication of the cell. So we start with a fibroblast, shown here in this very nice three-dimensional pseudo-colored rendering. This is a cell of the connective tissue. And you get two cells, two fibroblasts. In order for this to happen, the entire genome of the mother cell must be duplicated to produce two identical, or one hopes, identical daughter cells. Now, there is an interesting and noteworthy exception. There is one specific cell type in your body that replicates its DNA, but does not divide itself. And that's the sturdy hepatocyte, which is the cell of the liver. When its DNA is damaged, it does something very different from most cells. And what most cells do when their DNA is damaged, at least in you and I, is they undergo programmed cell death, apoptosis. Learn for life to pronounce this word with the second P silent, apoptosis. The hepatocyte, in contrast, doesn't have an option of dying because it is the body's defense against drugs, including drugs that damage DNA. So what it does is it replicates the DNA, and thus it becomes tetraploid, and then octaploid. Yes, folks, depending on what you did Friday night, there well might be some octaploid hepatocytes in your liver as we speak. I want to do everything I can to draw a connection in your minds between the rather dry narrative of mitosis. We'll spend 5 minutes talking about the metaphase to anaphase transition. Oh my goodness gracious me, be still our joint beating hearts, the metaphase to anaphase transition. I want to draw a connection between that and you, specifically that you are, of course, the product of mitosis. While, as I'm about to explain to you, most cells in you right now are mitotically quiescent, you, of course, started as a fertilized egg. And then you went through embryonic development, and embryonic development, or embryogenesis, is powered by mitosis. It's the mitosis in all its glory. From Professor Firestone in the third section of the class, you will learn quite a bit about the biological processes shown here, the early embryonic cleavages, and then, of course, pattern formation, lineage specification, the remarkable process of gastrulation. So I won't go into any of the biological details, but I do want to give you a preview of things to come. We have the good fortune of having here at Berkeley probably the world's leading authority on this process, Professor Richard Harland. And I wrote to him and I said, Richard, I'm going to lecture about mitosis in Bio 1. I'm wondering if you can share some movies. And voilà, mesdames et messieurs, I will show you some of Professor Harlan's work. I will first show you early development, early mitotic divisions in the Xenopus embryo. Xenopus is better known as the African clawed toad. They're very hardy and wonderful little, well, they're actually not little, they're kind of this big when adult. And they were used for pregnancy testing. And then they were just thrown out because it was widely assumed that they would die. They turned out to be marvelously hardy. So here you go, folks. Early division in Xenopus laevis. Okay. These are all one-cell embryos. They're going to start going through mitosis. You started this way as well. When you started, there weren't as many of other embryos around you, but you started this way as well. Now, what is happening right now is less visually spectacular, but biologically no less important. 3 major lineages of the embryo need to be specified: ectoderm, endoderm, and mesoderm, and that's happening as we speak. In particular, what I want to show you using a different species, the axolotl. Something that was essential to the fact that, for example, you formed a spinal cord and are thus able to have a central nervous system to listen to me and understand what I'm saying. Here you go, folks. After the blastula is formed, what the axolotl and the human being does. So you're now looking at quite a multicellular embryo. Check this out. This is a movie Professor Harlan took. There are hundreds of thousands of cells dividing and synchronously, beautifully moving to do this. You did that too. What is this? You will have to wait until Professor Firestone's section. But it is powered by mitosis. And most remarkably, as you are doubtless able to appreciate, one of the fascinating things about the process is, by definition, the cells generated in the mitotic process were non-identical. Some of them did this, whoop, and others didn't. So one of the truly marvelous things about the study of early embryonic development is our ability to understand how a process that in principle replicates the DNA perfectly, and in practice as well, yields cells that ultimately are non-identical. An adult human being consists of approximately 230 different cell types, all generated via mitosis. Okay. If you're ever in Madrid, I encourage you to go to the marvelous museum called the Prado and see this glorious work by Albrecht Dürer, the great German painter. It is, of course, Adam and Eve. And I can use this to remind you that in you right now, there's actually relatively little mitosis going on. Specifically, both in the ladies and in the gentlemen, in your bone marrow, your hematopoietic stem cells are dividing slowly. Their offspring are dividing faster to replenish, for example, the fact that your erythrocyte has a half-life, a full life of 120 days. So more red blood cells must be produced. Your intestinal epithelium renews itself. It takes about 6 days for a cell that arises at the bottom of the intestinal epithelium, concomitant with mitosis, to rise to the top, because the inside of the gut is just shed off. That's driven by mitosis. As all of you doubtless know, it takes about a month for your skin to more or less replace itself. So you go through many skins in your life. Now, in the gentlemen, but not in the ladies, a specialized cell type called the spermatogonia undergo mitosis at some time in your lives, and of course their progeny undergo meiosis. That doesn't happen in the ladies because, as most of you know, gametogenesis, or genesis, in mammalian females is over immediately after the onset of puberty. Mitosis is presented in your textbook in this rather narrative-laden schematic. My goal for the next 20 minutes is to both simplify it and illuminate it. So I will begin by removing all the verbiage from it, and then spend the next 20 minutes, or I don't know how long this is going to take, I'm going to spend some time walking you through what I'm expecting you to more or less memorize for the midterm. Because how cell division is controlled is one of those deep biological topics that people need to have a general sense of, irrespective of their future choice of career. With that, let us remove all of this text and begin with the obvious and the simple, namely that at the heart of the cell division cycle is, of course, the replication of DNA and the transfer of the newly replicated DNA to its daughter cells. Now, some of you will have the irresistible urge to fall asleep. Go ahead. You can watch this on webcast, but I will try very hard to wake you up. There will be another movie in a little while that I encourage you. And if your neighbor is asleep while the movie comes up, oh boy. One of the non-obvious things to people who study mitosis for the first time or the 40th time is it's very easy to do mitosis with one piece of DNA. Now, in fact, when human cells do mitosis, as I'm about to show you, there are how many pieces of DNA? Anybody know? How many pieces of DNA are there in the human nucleus about to go through mitosis? 46. Any other ideas? It's actually 92, and I'm about to explain why. But my point in walking you through the schematic is to highlight as strongly as I can that mitotic dynamics are invariant and independent of the number of individual pieces of DNA in the nucleus. Mitosis works identically whether or not there's one piece of DNA or 92. So we will focus on the cycle of just one and return at the end of the lecture towards what human cells do. But everything you have learned here will apply to what you did and are doing today. We start with one double-stranded DNA molecule represented by this silly little blue line with two lines. This is the Watson strand and this is the Crick strand. Let me emphasize. This is essentially a double helix. It's a very long one. So this is only 2 turns of DNA. This is 28 million turns of DNA, as I'm about to show you. But I do want to emphasize, this is a double helix. Okay? Now, the first thing that happens in the cell cycle is you have to replicate it, as we learned last time. All these glorious machines, the human version, of course, shows up to try to make an exact copy. And that takes a little bit of time, and I'm about to show you how much. But then it is done. So this molecule is replicated. And what do we end up with? 2. One more time. And I understand that most of you understand, but I really want to hammer perhaps is too strong a word. Place this into your cerebral cortex with force. Each one of these is a double helix. And they are identical to each other if the DNA replication machinery didn't make a mistake, and the DNA replication machinery in human cells doesn't make mistakes very often at all. Okay? DNA replication has been complete. The next thing that happens is actually quite remarkable, and we will talk a little bit about the implications of that process for the treatment of cancer at the very end of class if I have time. The next thing that happens is this DNA is compacted. As I'm about to tell you, the average human chromosome, the average human piece of DNA, is about 200 times longer than the cell it is in. So separating 2 of these very long things between the two daughter cells is a non-trivial act of spatial acrobatics. And the way Mother Nature has solved this challenge is by working out a way to compact this DNA from being 200 times the length of the cell to being actually shorter than the cell. DNA is compacted to form these 2 things. And then we have only one task remaining. We need to distribute this DNA to the daughter cells. Like so. And we will walk through the mechanics that drive that. And finally, we acquire two daughter cells, each with a copy of the DNA molecule that the mother cell started with. This is the general framework that I need you to more or less memorize. And you will say to me, look at, we learned this in kindergarten. I know, but I sort of want to refresh your kindergarten knowledge in your minds. The two organizationally most important slices of this pie chart, and this is of course partly why I went through the discussing with you Florence Nightingale's invention of the pie chart, is the part of the cell cycle when DNA is replicated and the part of the cell cycle when the DNA is partitioned between the daughters. The part of the cell cycle when DNA is replicated is known as the synthesis phase, or the S phase. There it is. The part of the cell cycle when DNA is divided between the daughters is called the mitosis phase, or the M phase. And the word mitosis has a very interesting Greek etymology that we shall get to in about 10 minutes. Now, I've already shown you this movie to its full length for a reason. I wanted to both inspire you, but I don't want to show it twice. So I've shown you this remarkable movie taken by Quorum Technologies, thanks again to them for providing me with this film, of early embryonic development in the Drosophila syncytium. One thing I didn't have time to mention when I showed you that film is early Drosophila development features a highly specialized form of the cell cycle called SM. Let me just show you briefly. I'm not going to spend all of 2 minutes showing you the film again. You've seen it. I'm just going to let the cells go through the first two divisions. I want you to count with your mind's math device the time it takes for this to occur, this phase in between, and the time that it takes for the cells to do this again. And what you see immediately is it's relatively fast, in that the cell doesn't take very much time between going through. This is because, as I've just told you, Drosophila embryogenesis employs a highly specialized cell cycle, SM. Let me show you in contrast, and you will see the contrast immediately, 24 hours in the life of a human embryonic stem cell. So a cell that is genetically kin to you and I. This movie was actually taken at the Whitehead Institute in Cambridge by somebody who got his PhD here at Berkeley, Dr. Ian Cheeseman. So the fluorescent blob is the nucleus, and this is a real-life human embryonic stem cell. And what is it doing? Well, it's mostly exploring its environment. This is time-lapse microscopy, and it's sort of going this way and going that way. Maybe it's time to start dividing. How long will mitosis take? And now I think this fellow down here is about to go through a pretty spectacular metaphase. What does this movie tell us? This movie tells us that the actual act of mitosis, which in this takes about an hour, occupies a relatively small temporal fraction of the overall cell cycle in dividing human cells. In fact, dividing human cells and pretty much cells in everything other than the Drosophila embryo spend most of their time not in mitosis, but in a part of the cell cycle called interphase. Not as an interface between two liquids, but PHASE. Specifically due to the addition of two relatively extensive gap periods, or gap phases. The first one is between the end of mitosis and the start of DNA synthesis, so gap 1 occurs between M phase and S phase. The second is between the end of DNA replication and the start of mitosis. So it's the gap between S phase and M phase. They're obviously not called first gap and second gap. They're called G1 and G2. G for gap. We can now distill this quasi-pie chart into the version that I'm hopeful will burn in your mind's eye for the rest of your life. Voila, the cell cycle. After mitosis is complete, the daughter cell enters the first gap, then begins replicating its DNA in S phase. Once replication is complete and two identical DNA molecules are produced, it enters the second gap, and then it decides to go into mitosis. It condenses its DNA to form two small things that are actually movable. It packs it up. It partitions via the process of mitosis in M phase the two chromosomes between the two daughters, and the process can repeat itself. I am expecting you to be able to narrate this to your baby sister. Like literally, if you have a younger sibling, call him or her and say, I'm going to tell you about the cell cycle. And practice this narrative. Seriously. If you don't have a sibling, use a roommate, a significant other, your parents. I don't care. Practice. Narrate the cell cycle. If they hang up the phone, find somebody else. In Adam, Eve, and all the gentlemen and ladies in this room, the majority of the cells, in fact, are in none of the four phases of the cell cycle that I just showed you. In fact, the majority of the cells in our bodies are in a specialized metabolic and non-mitotic state called G0. I will teach an entire lecture about the regulation of the cell cycle about a week and a half from now. And we will go back quite a bit to the schematics I just showed you. But the irony of this rather large focus on mitosis is that, for example, the cells with which you are understanding this, your neurons in your cerebral cortex, are actually not mitotic at all. They are completely terminally post-mitotic. So be nice to yourselves. Ladies and gentlemen, we now begin one of biological instructions best known, cures for the will to live. The phases of mitosis, which I am expecting you to please memorize. They are strangely named, and the partition between them comes across as slightly arbitrary. And I empathize with your predicament. So there is interphase, prophase, prometaphase, metaphase, telophase, anaphase. Did anybody notice I inverted the order? Good. Then there's passing phase, solid phase, in your face. No, there isn't. My goal over the next 10 minutes or so is to shine a spotlight on each of these awkwardly named phases. What they are called is irrelevant, even though you are required to know them. What matters is what happens in them and what the evolutionary logic is of that taking place. With that, by the way, there is no in your face. It's a joke. If I were to ask you, and please don't shout because there is no correct answer. This is a trick question. What is this? No, don't say anything. It's easier for me to tell you what it is rather than what it's called, because what it's called is actually really complicated. Okay, well, what is this? It's two pieces of DNA. Well, what do biologists call that? Do they call that two pieces of DNA? Ah, this is when the sadness begins. Prepare for the sadness. This is a gorgeous picture. It is taken by using a technique called spectral karyotyping, and it was taken by Dr. Thomas Reed of the National Cancer Institute, and he graciously provided it to me. This is a metaphase spread. Physicians, scientists, public health professionals in the room are expected to know that phrase. The metaphase spread. It's essentially a way to look at the human karyotype. Before you folks are 92 pieces of DNA. This is how many pieces of DNA lie in the human nucleus at the metaphase to anaphase transition. 46 pieces, 2 identical copies each. They are numbered according to size. The largest one is 1. This is 2 pieces of DNA. And this is 2 pieces of DNA. Then there's 2, and so on and so on. This is not Photoshop, by the way. They're visualized by hybridization with probes that glow different colors. So you can actually identify that. I'm actually partly colorblind. So to me, these colors look the same. But I'm hopeful to most of you they look slightly different. I need to walk you through a little bit of the sadness of the nomenclature before we can dive into the phases of mitosis. A human genome contains 33,000 genes. Now, let me emphasize that counting genes is a little bit like counting stars. It's fun for some, but not all. And from my perspective, it's somewhat missing the best part. So there is an extended polemic how many genes the human genome contains. I give you my word, it is not the interesting bit. Stay away from it, focus on something else. What's a gene? There are actually several definitions, depending on whom you talk to. For purposes of right now, I'm going to simply tell you that it's a DNA-encoded instruction to build a protein. There are 19,000 of these. Again, notice this footnote. Or to do something else, the rest. And you'll say to me, well, thanks for teaching us absolutely nothing about this. Don't worry, there will be something about this later. Those genes are actually distributed between 23 pieces of DNA. And geneticists, those who study how the genetic material work, call each of those pieces of DNA inside your and my cells a chromosome. Now, a few caveats. There's one important gene on another piece of DNA called the Y chromosome. The gene is called, it has a fantastic name, it's called sex reversal on the Y. Okay, so let's make that 24 pieces of DNA. Oh, and of course mitochondria contain their own genome, which is tiny, but it's there. Okay, let's make that 25 pieces of DNA. Done. I need to give you a specific example because I don't want this to be disconnected from what I talked to you earlier. You will remember the boy who was cured of his blindness by injection of a piece of DNA that rescued his retina from degeneration. The mutant gene that he carries actually lies on chromosome 1. Human chromosome 1 is 250 million base pairs long. It's 7% of the entire genome. It contains, depending on how you count and whom you ask, 2,100 genes. 69 million base pairs from its end lies the RPE65 gene that I've told you about, mutations in which cause Leber's congenital amauropathy. Note, the word chromosome really isn't necessary. We could simply say piece of DNA, because that's what a chromosome is, a very, very, very long piece of DNA. Now, don't for an attosecond think that this is an accurate representation of this, because it's not. There are 10.5 base pairs per turn of the double helix, so chromosome 1 has 23.8 million complete turns of the double helix. The drawing above has 13. In 1879, right around the time when Friedrich Nietzsche in Basel was purifying DNA from pus, this gentleman, who looks rather majestic, I have to admit, his name is Walter Flemming, published a remarkable paper, which I like for 3 reasons. First, it has the world's best word, Lebenserscheinung. It is German for those aspects of the appearance of the cell that have to do with the fact that it is alive. German is a fantastic language. Second, because of how understated the rest of the title of the paper is, contributions to knowledge about the cell and of aspects of its appearance that have to do with the fact that it is alive. This paper describes mitosis. So instead of saying the single most epic discovery in the history of biology up to the present moment, he goes, contributions to aspects of the knowledge of the cell. Oh. Folks, I give you the first time that humanity saw mitosis in Walter Flemming's remarkable 1879 paper. These are his own drawings, and think about the fact that electricity was not very common back then, putting it very mildly. So he didn't exactly have high-resolution microscopes. So if you're ever in VLSB, there's a beautiful exhibit of microscopes. Find one that was contemporary to Fleming and imagine drawing these. Fleming saw that dividing cells appear to contain threads, these things. And mitos is Greek for thread. So he decided to call what he just discovered mitosis, the process with threads. By the way, are these human cells? No. They come from the California slender salamander, which lives in Berkeley. It does. It's a California and, in fact, a Bay Area native. So it is an objective fact of the science of history that this beautiful little critter made this indelible contribution to the history of science. Mitosis was discovered in it. Why in the California slender salamander as opposed to, I don't know, the large German warthog? Well, because the salamander has very large chromosomes. They're easily visualized. The last reason that I, and everybody else pretty much, admires Fleming is he was able to correctly determine the stages of mitosis. What you start with, what you go through, and what you end with. But folks, as he was observing preparations of salamander cells, those cells were dividing asynchronously. It's not like you could take the salamander and say, this group of cells begin mitosis. In other words, he would see a cell that looks like this, and then he would see a cell that looks like that, and then he would see a cell that looks like this, and he needed to correctly put them in order of biological appearance. He did that, which is a majestic accomplishment. He did not coin the word chromosome. It appeared very shortly after his paper came out. But bottom line is, the world owes the word chromosome to Fleming's brilliant work. Today, geneticists use the word chromosome to mean a piece of DNA inside the nucleus of a cell with a bunch of genes on it. The word was coined by cytologists, who are brilliant scientists who look at cells, but the point is this was done 75 years before discovering the structure of DNA, and at least 30 years before discovering that inside chromosomes is DNA, which is the genetic material. So very soon after coming up with the word chromosome, cytologists realized that in dividing cells, if you squish them just the right way, you can see this or that. Cytologists then proceeded to make up another word. In retrospect, we know this wasn't necessary, but there's a Russian proverb that translated goes, if I knew where I was about to fall, I would put some hay there to soften the landing. Which is the Russian version of hindsight is 20/20. If I could jump into a time machine and go back and say, don't invent the word chromatid, but it's too late. Enter the chromatid. It was made up to explain the fact that during mitosis, a chromosome squished the right way appears to generate an X-shaped structure. And this is a picture from your book, February or whatever, March 2012. Chromatid was thus coined by way of a name for one half of this X structure. So there is a chromatid and there is a chromatid. In a stunning example of bad luck that will haunt biology for the rest of its existence, the term chosen for each chromatid that forms a chromosome is sister chromatid. So, folks, I give you sister chromatid number 1, and I give you sister chromatid number 2. Other than Sauron or Voland, this literally was the worst possible choice of name. And let me explain why. By show of hands, I don't suppose any of you by some miracle know who these are. Please. The Bronte sisters, good for you, I'm very impressed. Charlotte, Emily, and Anne, the authors of Jane Eyre, Wuthering Heights, and other classics of English literature. Now, I'm very impressed with the fact that you knew this. Incidentally, if I had shown you this picture and asked you who these are, the irony is millions and millions of people knew who those people are, largely because millions and millions of people know who these people are. But these people are not just known, they wrote amazing books. It's a sad testament to the way our world works, that being known is a reason for being known. They are related, but they are not genetically identical, as all sisters are. the technical term for two human beings who are largely genetically identical due to the identity of their DNA is twin. This is from the current National Geographic. There are some twins. There are more twins. After DNA replication, two identical twin pieces of DNA remained attached to each other via a set of proteins. This is the biological truth. Each of these pieces is now called a sister chromatid. So we are told that together they form a metaphase chromosome. For example, when human chromosome 1, a long piece of DNA with the RP65 gene on it, replicates in S phase, it generates two sister chromatids. But they are identical to each other, and they are identical pieces of DNA. Each one is human chromosome number 1. Each one has the same version of the RP65 gene and all other genes. Thus, the term sister chromatid is first unnecessary. Second, it uses the wrong noun. They're not sisters, they're twins. But there's nothing we can do. This is what you need to know. But I did want to make sure that you understand the underlying biology. Now, there are specific fields of study where using the word chromatid is useful, and we will talk about them a little bit. But to people taking Bio 1 and just excited about the future, feel free to stay away from it after you're done with taking the midterm. So the function of mitosis then is to provide each of the daughter cells with a copy of the genome identical to that of the mother. So mitosis needs to, depending on how you want to state this, parse the sister chromatids into two daughter cells, or what I like better is parse the two identical pieces of DNA into two daughter cells. And this is not a small task. A human nucleus in G2 contains, as I said, 92 pieces of DNA, 23 pieces of DNA chromosomes from mom, 23 from dad, and each one freshly replicated. So I took this figure from your book, I stripped it of all its text, and added only the things that I need you to know. G1. The cell prepares biochemically for the rather majestic act of replicating its entire genome. There is a lot of self-doubt. Should we be doing this, is what the cell is asking itself. And in the lecture on cell cycle control, I'll walk you through the mechanisms of that self-doubt in considerable detail. This arrow, of course, designates the no small feat of replicating the entire human genome, 3.3 times 10 to the 9th base pairs. Think back to the analogy I put with the stack of textbooks, 10 of me tall. So, an identical copy of that is made. And we enter G2. DNA replication is complete. Each chromosome now exists in two identical copies. And they remain attached to each other, not emotionally, physically, but not for long, as you shall see. Okay? Mitosis can now begin in earnest with something called the prophase. In prophase, chromosomes condense. Notice that each X shape is two identical pieces of DNA. The mitotic spindle, a remarkable cytoskeletal machine, begins to form. And I will show you a movie of the mitotic spindle very shortly. Prometaphase. The nuclear envelope breaks down. Microtubules from opposite spindle poles attach to kinetochores. This is, for now, let's call the kinetochore the constriction in the middle of each chromosome. Now, I really should have said of each pair of chromosomes, right? Because we know by now that there are two pieces of DNA inside there. But fine, let's use the word chromosome. As I mentioned to you, the MCB department has the wonderful advantage of having world leaders of study of essentially every aspect of biology. I mentioned to Professor Rebecca Heald of the MCB department that I'm going to talk to the students about mitosis, and she shared with me this marvelous alternative view of early division in the Drosophila syncytium. Remember the very chromosome-centric view when it goes shook? Let me show you a mitotic spindle-centric view from Professor Heald. The cytoskeleton is in green, the DNA is in blue. and again, and yeah. That's remarkable. That the glorious machinery of DNA replication that I showed you earlier and the amazing mechanism for condensing chromosomes is accompanied by no less an amazing machine for making sure they're separated, the mitotic spindle. Done. Okay. Okay, let me walk you through the schematic version thereof. Folks, we now enter metaphase. Chromosomes have aligned at the spindle midpoint, and the cell now, and this is actually truly wonderful, you can actually show experimentally that this is true. The cell will now wait until all the chromosomes are attached to the spindle, and correctly so. What does that mean? Remember, the goal of this process is to give the daughters identical genetic dowry. The way you actually affect that is making sure that of the two identical copies of chromosome 1, one goes to the left cell and one goes to the right cell. And as I'll explain in the lecture on regulation, a component of the mechanism is making sure that each freshly replicated chromosome, two chromosomes lying side by side, is perfectly, correctly attached to the mitotic spindle, the way you see here. Because if each one is attached correctly, then the cell will have no problem sending this copy of chromosome 1 this way and this copy of chromosome 1 that way. And the cell waits, sort of. So all of those motions of the chromosomes on the metaphase plate have to do with waiting and the spindle and the chromosomes talking to each other. Is everyone in place? Are we lined up? Are we good? Because as I'll describe to you a little bit in the lecture on cancer, or early embryonic development as well, if a mistake is made in parsing the genetic material between the two daughters, all sorts of unfortunate things can transpire. But once that is done, you have this inspiring moment, the metaphase to anaphase transition. This is when the chromosomes go, wah. The spindle, of course, separates the two sister chromatids, moves them to opposite spindle poles. This is the narrative in your textbook. In English, two identical pieces of DNA now become separated and move into what will become two separate cells. And we're more or less done. Hence telophase, telos, the end, telomere, teleology. Chromosomes return to their interphase state, which is decondensed. The new nuclear envelope will form, and the cytoplasm will begin to divide by furrowing. Please. Oh, thank you for asking. This lady is asking, do the chromosomes move to the spindle fibers, to the edges of the cells? Or is there, in other words, does the chromosome act like a Pac-Man and go like this? Or do the edges of the spindle go like that? I will literally spend 10 minutes on this in the lecture on cell cycle control. I don't want to do injustice to the nuanced answer. Good thinking. One wonderful historic example, and then we will be more or less done. Prometheus was one of the Titans, and he stole fire from the gods and gave it to humans so that we could warm ourselves up. Anybody know how the gods punished Prometheus?
They tied him to a mountain and had an eagle hack at his liver all day. It would go back overnight and the liver.
Correct. This gentleman is correct. They tied Prometheus to a mountain, and then this eagle would show up and eat half of Prometheus's liver. And then the liver would grow back. The reason I bring this up is, whether true or not, this narrative illustrates an important biological point. The human liver has a remarkable capacity for regeneration after hepatectomy. So quiescent hepatocytes, mitotically quiescent, sitting in the liver, can re-enter the cell cycle. On command, the bird has, oh God, that bird again. Fine. And regrow the liver. So regulation of the cell cycle is a process of marvelous biological intricacy and nuance. And I will talk to you about it in considerable length. The quiz. You were hopeful there wouldn't be one, did you? So I deliberately structured the quiz to make sure that it connects to what I'm about to say next, which I may or may not have time for, but at least You know for yourselves that you did not trek all the way here for nothing. First of all, let me launch. All right, folks, as again, the format is the same, and I promised you I would ask you about this, et voilà. One of these statements about DNA replication is wrongity, wrong, wrong. Which one? A, DNA must be unwound before it can be replicated, because synthesis of the nascent strand requires Watson-Crick base pairing between the incoming DNTP and the nucleotide in the template strand. B, DNA polymerase cannot initiate replication without a primer. This primase must first provide that primer, which in fact is made of RNA. C, the terms leading strand and lagging strand refer to the strand on which DNA synthesis occurs in the 5' to 3' direction, and to the one where it occurs in the 3' to 5' direction, respectively. D, Okazaki fragments are found exclusively on the lagging strand. And E, the bagel-like structure attached to DNA polymerase is a clamp, the biological function of which is to ensure high processivity of the enzyme. Let me move the sensor to the center so that you can actually transmit. All right, please go ahead and take 90 seconds to pick the answer. Talk to your neighbors, look at your notes, Google this issue, post the question on Facebook, anything. Everything goes. All right, folks, another 30 seconds, please. 25 seconds. You can do this. You know the answer is 42 if you don't know what the answer is. Come on. Almost there. You can do this. 10 seconds. 10, 9, 8, 6, 7, something or other. Can't count backwards. 3, 2, 1. All right. And bravo. Good job. Good for you. You learned this. I'm very, very happy, and you should be happy too. And I don't have time to tell you about anything else, so see you Friday.